Hello, everybody. Today we will be talking about nerve histology, mainly the two questions of types of nerve fibers, their sheaths and endings, neuroglia, common features, classification, and function. Let's start. So the cells of the nerve tissue are developed from the neuroectoderm. They are also the main components of CNS and PNS, and they can be classified as neurons and glial cells. The way that they spread information towards each and every neuron are both electrically and chemically. This is through action potentials and neurotransmitters. Now, the neurons can be split into three categories based on their function or functionality. So we have motor neurons, we have sensory neurons, and we have interneurons. So motor neurons are efferent function, so they go away from the CNS. An example would be that the neuron would go into our muscle fiber, and an action potential would go through that neuron into our muscle fiber, and that is basically how we can contract our muscles. Our sensory neurons are afferent in function, and so they go towards the CNS. An example could be that there is cutaneous sensation from your skin, and so that information is obviously sent towards the brain. And we also have the interneuron, which connects these two. Um, and if you would to see all three in action, you would have, for example, a reflex arc, but that is not today's topic. It's just an example to understand more about it. But the main topic we will talk about today are the myelinated nerve fibers and the unmyelinated nerve fibers. So the myelinated nerve fibers are basically covered by myelin sheath, and they basically cause the impulse conduction to be very rapid. And this is because they are insulated, and because they have a function of insulation, this causes the ions to not leak out of the nerve. And so this basically causes an efficient impulse conduction to happen. And between CNS and PNS, the myelin is produced by different cells. So in CNS, it is produced by oligodendrocytes, and by PNS, it is formed by Schwann cells. Um, and we will talk about more about their differences in later slides. Just an extra information about the histological slides of myelin. Because myelin contains an abundant amount of lipid, during the procedures of, like, for example, basic histological processes for HE, for example, we have a process known as embedding process, which basically uses paraffin wax to solidify like the tissue. So we can, split the tissue into different slides, into different layers. Myelin is very myelin contains lipid, and lipids are very soluble in paraffin wax. So that's why, for example, in HE, they would not be seen sometimes, or they would not be seen at all, actually. And between adjacent myelin sheath complexes, we have gaps known as nodes of Ranvier, and they are very heavily concentrated in voltage sodium gated channels. And because of that, this basically causes the action potential to keep on going, because there is always continuous exchange of ion due to the continuous process of sodium channels exchanging its ions with potassium due to the presence of node of Ranvier. And this can give it its name as saltatory conduction, because it makes it look like as if the impulse is jumping from one node to another. And unlike oligodendrocytes of the CNS, the Schwann cells only wrap around one portion of one axon, whereas the oligodendrocytes will form many different foot processes with many nerves and many axons. We will see later. Um, Unmyelinated nerve fibers are in CNS and PNS as well. In CNS, however, it is very prevalent because CNS, it has a lot of function with, for example, in our cerebral cortex, it has a lot of function of regulating or regulation of information. So it would make sense why most of it would be unmyelinated. In PNS, it is not wrapped around the Schwann cells. However, it is still covered by the Schwann cells. So you could say that it is covered by a glial sheath, because Schwann cells are known as glial cells as well. And because they are unmyelinated, their impulse are conducted slowly. And yeah, here we have two schemas of PNS and CNS sheaths, myelin sheaths. So in PNS, we can see that the Schwann cell wraps around the axon multiple times with one axon only in one portion. And on the right, we can see the oligodendrocytes wrapping around multiple processes and multiple nerves and multiple axons with just one cell. And more information of myelin sheath and PNS. We have the nodes of Ranvier, like I said. It's the unmyelinated part of the axon, contains an abundant amount of voltage-gated sodium channels. And we also have the internodes, which are basically the myelinated part of the axon. It's just a fancy way of saying it. And next we have something known as the Schmidt-Lenterman clefts. And what that is basically it's like a packet of they're like packets of cytoplasm that are left behind during Schwann cell forming into myelin. So they're basically just remnants of that. And yeah, that's basically it. And we can also see it in more detail in many different concentric layers. So going from inner towards outer, we have the nerve fibers, the nerve fascicles, which contains a bundle of nerve fibers, and then we have our nerve ultimately. Our nerve fibers are covered by endomyrium, which is loose connective tissue, and our nerve fascicle is covered by flattened cells. However, you can also say that they are covered by flattened fibrocytes, and it will be mentioned in other videos that fibrocytes are basically non-functional fibroblasts, and they also contain tight junctions, which are known for ion exchange. They are also covered by specialized CT, which basically helps in the blood-nerve barrier, which separates the nerve and the blood vessels, as you can see in the diagram. The nerve is also covered in the end by the epineurium, which contains dense, irregular connective tissue. So we also have, I also put some demyelinating diseases in the slide just to show you how important myelination is for nerves in both CNS and PNS. In CNS, we have multiple sclerosis, and in PNS, we have Guillain-Barré syndrome. The way that they are caused is different, but the symptoms of it are somewhat similar because they both contain, for example, lack of muscle coordination, loss of cutaneous sensation, and so on. So yeah, just important to note. And nerve endings. So basically we have three types: sensory nerve endings, motor nerve endings, and autonomic nerve endings. And based on function, they can be excitatory or inhibitory, depending on what neurotransmitters are secreted. But we will mainly be talking about motor nerve endings and the motor end plate. So basically, it's the nerve that is coming in contact with the muscle fiber. But I will not be explaining the mechanism of it. I will be explaining what it has to do with the Schwann cell and everything like that. So again, the axon loses its myelin sheaths and it basically widens to basically close its contact with the muscle fiber. And ultimately, the Schwann cell's external lamina will fuse with that of the sarcolemma. And as you can see here, the sarcolemma does not contain the myofibrils. The myofibrils are down, like more down. And the C level in the picture, you can see the letter C, that's basically where the myofibrils are. But in the sarcolemma, it basically just contains the mitochondria and the T tubules used to basically spread the action potential. And yeah, here is just another picture of the motor end plate through SEM. And here are just a couple of neurotransmitters. Like I said, we mentioned acetylcholine, which stimulates the muscle contraction involved in both PNS and CNS, catecholamines, serotonin, amino acids, and they all can either be both inhibitory or excitatory. Now let's look at the slides of the peripheral nerve. So here we have a cross section. Like I mentioned before, some histological staining can remove the myelin due to the lipid nature of myelin. But here in Luxo Blue, we can clearly see that myelin is clearly visible due to the staining method or the procedure. And over here in the slide, you can see there's many layers like the epineurium, the perineurium, and you can also see the nerve fascicle and bundle. Here's just another part, another view. However, it's just a different staining. Like I said, HE, which uses paraffin wax, so you won't see the myelin here, but you would know where it is because it's it's obvious. You can also see the nodes of Ranvier, and you can also see the axon in the cross section. In the longitudinal, you can also see the Schwann cell nucleus as well. And yeah, here is just another section. You can just look at it. And now let's talk about glial cells. So in general, what they all have in common is that they support the cells of neurons, whether that is through nutrition, protection, defense, or basically maintaining the microenvironment of that neuron. So in CNS and PNS, there are different types of glial cells. For example, in CNS, we have astrocytes, oligodendrocytes, microglia, and ependymal cells. In PNS, we have Schwann cells and satellite cells, and we will get more into it. So astrocytes are two types. There are protoplasmic astrocytes and fibrous astrocytes. The difference is that the protoplasmic astrocytes have many branching processes, uh, and they are also um, mainly seen in gray matter. In fibrous astrocytes, it is containing fewer processes, and it is mainly seen in white matter. But in general, the foot processes of the astrocytes, they will attach towards the pyometer, the surface of the nerve fibers, and the wall of the vessels. And when we talk about the wall of the vessels, we talk about the BBB, or the blood-brain barrier, which is important because it helps in the exchange of nutrients and wastes and all that. And in general, they basically have a supporting role essential for survival and maintaining the ionic environment of the neurons. And when you think about astrocytes, in the next slide, yeah, when you think about astro in the beginning, think about star-like shapes. So in each of these images, you can see that they are star-like shapes, like astrophysics, study of stars or space. And yeah, what astrocytes also contain are GFAP, which are glial fibrillary acidic protein, and they are basically the intermediate filaments of the astrocytes, and they are mainly seen in astrocytes. And what they are basically used for is that they are basically mainly used to detect the glial cells of the astrocytes. All right, so here we have an oligodendrocyte. Like I said already, one oligodendrocyte can myelinate several nerve fibers, and the slide over here, it sort of resembles like an egg. So whenever you see these type of like egg-like structures, then just know that they are always oligodendrocytes, and they are derived from the neuroectoderm. And now we have the microglia, which are mesenchymal in origin. They are basically derived from the bone marrow monocyte precursor cell, and they helped in phagocytotic activity of the CNS, whether that is due to injury or infection. And they are antigen presenting, so they are very important in immune function. And they also form ROS, which are reactive oxygen species, which also help in fighting infection and phagocytoting. However, in several diseases like ALS, there seems to be a problem with the ROS and the microglia, so that's just something important to note. Like I said, they are mainly used for CNS phagocytosis, and they also elicit inflammatory responses by producing inflammatory cytokines. And next we have the ependymal cells, which basically can range from cuboidal columnar to simple cuboidal. Um, and basically, when you think about cuboidal, you always have to think about fluid regulation. So that is basically what its function is. And they also contain cilia and microvilli to also help absorb the CSF or the cerebral spinal fluid. And they also contain tight junctions. They also do not contain basal lamina because basal lamina, to recall, is basically used for structural support. So with the function of the ependymal cells, it would make sense for that to be there. and they are also used for the production of cerebral spinal fluid, for example, in the choroid plexus. Here is just another staining slide of the epidermal cells. Here we have Nissl substance and He. And lastly, we have the PNS glial cells, which are Schwann cells and satellite cells. Schwann cells, we explained before, they're basically uh, producing the myelin for PNS, and they only myelinate one axon of one portion. And satellite cells, they're they're also enveloping the cell body in the ganglia of the PNS, and they are used for maintaining the microenvironment and also used for metabolic exchanges like waste and nutrition. And just another thing to note, like I said, oligodendrocytes are derived from the neuroectoderm, and so are other microglia. So basically, so are other glial cells. So like astrocytes, oligodendrocytes, microglia, ependymal cells, they are all from the neuroectoderm. However, it is the Schwann cells that are the most different because the Schwann cells are exclusively derived from the neural crest. So that is just something important to note. Yeah, that is it, and thank you for listening.